Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Hi everyone, Patrick here. Before we get into today's episode, I just wanted to let you know that we have just welcomed a new baby into the family, so I'll be on paternity leave for the next couple of weeks. While I'm away, we decided to rerun some of our favorite episodes from the archive. These are some timeless lessons from some really amazing people that I think you'll enjoy whether you're watching them for the first time or revisiting. So I'll be back with new episodes and new guests in a couple of weeks, but until then, thank you for listening and see you soon. Welcome everyone to the Genetics Podcast. I'm really excited to be here today with Sal Rico, who's the Chief Medical Officer of Encoded Therapeutics, and he has spent more than a decade in gene therapy, and prior to that, I think a decade in drug development more broadly. We're going to talk through Sal's background. He's got a couple of really interesting stories of how he got to where he is today. But also we're going to do a deep dive into gene therapy in neurological diseases, in particular in children, where Sal has spent a lot of his time and career recently. So I think it's going to be a really informative episode for everybody to learn where we are at the cutting edge and some lessons, I think, some very hard won and fought over the last few years that Sal's been working. So thank you for joining me here in person as well. It's really nice to do this in person.
Indeed. Thank you so much, Patrick, for the invitation. And thank you so much for doing what you do. I think that, especially in this day and age of misinformation, it's very important that we have sources of great information like the one that you get from multiple experts in the field. So thank you for giving me the opportunity to be here with you today. And I agree with you. Doing this face-to-face is priceless.
Yeah, I love the podcast format because you can go really deep. You don't have to try to distill something down to 140 characters or whatever it is and hope that it makes some sense. You can really go deep. And with that in mind, I'd love to start actually with how you got into developing therapeutics from the beginning. This wasn't something that you thought of when you were in high school or college and said, I'm going to be a drug developer. It came a little bit later through some direct experience you had. I wonder if you could take us back there and how that came about.
Absolutely happy to do that. I think that I am, like many people, the product of my upbringing, curiosity, hard work, and the influence of many, many people along the way. So as I think about my path, I think that there are distinct points, turning points that I've identified that led me to work now in drug development and specifically in gene therapy. The first one was when I decided to go into medical school. Then the second one was when I decided to go into clinical pharmacology, and then more recently into gene therapy. So if I go back to my last year of high school in Mexico, where I was born and was raised, at the time, I was trying to decide between going into law school, medical school, and computer science. So very different. And I think that my father would have liked me to become a lawyer. And my mother, who's an anesthetist, perhaps would have wanted me to go into medical school. So I think that she was happy with my choice. And I was reasonably good with computers back then. And I decided to go into medicine, not only because I was interested in biology and interested about diseases, and I was always fascinated by my mother's stories of the hospital, but also because I thought to myself, when I'm 40-something years old, I don't want to spend my day staring at a computer screen. And the irony is not lost on me because that's what I do now 80% of the time. But then I went into medical school at the National University, Autonomous University of Mexico, had a great time. The second year we had a strike. And I think that that was an important turning point because during that strike, I wanted to do something with my time. And I volunteered at a clinical pharmacology unit that was part of the National University. So that was my first experience getting closer to the drug development process. And then I got even closer during my internship in Madrid, where I went to a clinical pharmacology unit as a research volunteer because I wanted to get some money to travel in Europe.
Oh, yeah.
And so that was a second point of interest. So later, as I was completing my training, I decided that I wanted to, enroll in a postgraduate diploma in clinical pharmacology. And it was at the end of it when I thought that that's what I wanted to do and that I wanted to go into industry and lead the development of multiple therapies for different diseases. And one of the things that I did back then is I identified about 100 individuals in industry, chief medical officers, development, and I sent them an email introducing myself and asking them for guidance. I'm a young man trying to look for guidance. How did you get there? How did you go from becoming a dermatologist to being the CMO of a company working in neurology? Right? I was fascinated by the tours. And about 10 of them were very, very kind and very responded to me. And I got on the phone with a couple of them. And at the end of that exercise, I decided that I needed to pursue a PhD in pharmacology with clinical emphasis. But before doing so, I wanted to get a little bit of hands-on experience and better understanding of the drug development process. And I also needed to identify the program and get funding for it, very importantly. So at the time, I joined a contract research organization, an American company that had an office in Mexico. And they had an opportunity, both as a medical monitor and in clinical operations, that really gave me the foundation of two important pillars in clinical development, and that to this day I use and I understand now what my colleagues are doing in those departments. So then eventually I did find a program in Barcelona, the Drug Research Center at St. Paul's Hospital. I went there thanks to a scholarship from the Mexican government. And there I designed, conducted, and analyzed many different clinical trials, a lot of first-time human clinical trials, a lot of phase 2 and phase 3 clinical trials in coordination with the departments of the hospital. And my mentor then also pushed me to start consulting for a few companies, both in Europe and also some of them in the US. And thanks to a now mentor and friend who I had met during my days at the CRO, I also started consulting for a company called Anacar Pharmaceuticals in Palo Alto that had spun out of Stanford, that had a Boron-based library that was being utilized to develop antivirals, antibiotics, and some anti-inflammatory agents for dermatological indications. This person, Dr. Buettner, also eventually introduced me to the leadership of Transfer Pharmaceuticals. So as I was completing my program and I was working as a researcher at the Drug Research Center, it was Transcept that initially offered me a consulting gig, and that eventually became more serious. And to the end of 2010, they asked me if I would relocate and lead their clinical research team. And at the time, I was really excited to go to a biotech hub, such as the San Francisco Bay Area. So I joined. It was a fascinating time there. I contributed to the development of middle-of-the-night insomnia drug. We got 2 complete response letters from the FDA. So it was some tough lessons on regulatory interactions back then with the FDA. Some also interesting business development lessons because that drug was licensed to Purdue Pharma. But since it took us a little bit longer to get it to approval, by the time it got approved, Purdue was already being challenged by some of the lawsuits as a result of the opioid epidemic. So they couldn't really sell the drug that we had licensed to them. And it didn't help that our second program was in treatment-resistant obsessive compulsive disorder. And we had in our phase 3 program a very high placebo response rate, 60%. So although we had some of the programs earlier in the pipeline in migraine and opioid dependence, we became rapidly the target of some activist investors. And the company had to do a reverse merger. So at that point, I made a transition to another company called Cirrus Corporation to work on pathogen and leukocyte inactivation for blood components. This combined a drug and a medical device to treat blood components such as platelets, plasma, and red blood cells. And I didn't know anything about transfusion medicine, and I didn't know anything about many of the indications for which these were being used. And I didn't have experience with medical device development. So I thought that was a very interesting skill set that I could acquire as a developer. So I joined, and my time there was exhilarating. I contributed to the approval of 2 of the products, so for plasma and platelets. I conducted trials in cardiac surgery, thalassemia, hematology oncology, and also a couple of expanded use trials as a result of Zika and chikungunya and dengue virus epidemics. So I was very happy there.
We were busy.
Very busy. Yeah, it was a great time. But then at the end of 2016, one of my former colleagues from Transept sent me this groundbreaking breaking paper on the use of AAV gene therapy for the treatment of dogs with myotubular myopathy. So myotubular myopathy is the result of a mutation in the MTM1 gene that encodes for myotubularin, that is a protein that is fundamental for the normal development and function of muscle cells. And without this protein, these dogs rapidly became very weak. They lost mobility and they eventually were not able to breathe or swallow. So they prematurely died around 12 to 16 weeks of age. And a single administration of this drug had incredible results, rescuing the mortality associated with the disease. But not only that, you could see videos of the dogs that could not move at one moment, and then a couple of weeks after the administration, you could actually see how they were moving, and within a couple of weeks more, they were running around. So although I had no experience in rare disease drug development and had no experience in gene therapy, I thought this is really transformative. I did have over a decade of experience with small molecules and medical devices. So I knew that this was revolutionary. It sounded like science fiction. And I just wanted to be part of it, wanted to be a part of that story. So that's when I joined the gene therapy realm, and I haven't looked back since.
We asked you to share just a couple of notes ahead of the call, and one of the things that that stood out to me that you mentioned is someone probably around that time told you if you get into genetics, then you just should be prepared for a lot of frustration. Has that been accurate? There's probably been lots of highs and lows, but when you get in on the ground floor of something like that, especially as new as gene therapy, you're bound to have some challenges that you just don't have in small molecule world, right? What were some of the big highs and lows over the last decade there?
Yeah, absolutely. This was back in 2000, so 25 years ago.
Oh, wow.
This was my first clinical genetics professor. It was my first class. And one of the things that she said, she gave us this stark warning that don't go into genetics unless you have an extreme tolerance for frustration. And you know what? Back then, she was completely right. 25 years ago, clinical genetics just was filled with dead ends from a diagnostic perspective and very few things that you could offer to those families and those children living with the disease. At best, you could sometimes offer them a diagnosis, and sometimes not even that. So I completely agreed with her back then. And now, as I think about what we have accomplished and some of the approvals that have occurred over the past 10 years with these genetic medicines, I cannot but feel fortunate to be part of writing a chapter in the development of new therapies that are changing the way medicine is practiced already. And I would say that this is barely the beginning of that revolution. So, but at the same time, I think that the frustration now comes from other sources. I think that now the technology is there. I think that now, and especially ever since CRISPR came about and many of these gene therapies have gotten approved, I think that now we have questions such as, how can we go faster? How can we reach more patients? How can we go from monogenic disorders to polygenic disorders? How can we make access to some of these therapies more equitable so that once you get them approved, you actually can use them in all the patients who need them, right? So the frustrations come from a different place. I think that the other part of the frustration is that things take time and things are not easy, and biology is at times treacherous, and things may end up being more complex than what we appreciate. So there have been a lot of learnings along the way that I think that are helpful for us. But again, I think that on the one hand, it's very rewarding to see some of these advances becoming a reality. On the other hand, we do face certain frustrations in spite of some of the breakthroughs that we experience.
Yeah, I want to talk a little bit about what you're working on at Encoded Therapeutics right now. But before we go on that, I want to just talk a little bit about what is probably, I mean, you tell me how you feel about it a few years on, but a harsh lesson I think that you had from the time that you were leading XLMTM development at Audentes. You talked about XLMTM therapeutics in dogs and the amazing videos that you saw. And I've been at enough gene therapy conferences to see those videos in kids. now in all kinds of different disorders. And you spent about 3 years of your life working on gene therapy for XLMTM. I read the paper in quite a bit of detail. And what is striking about it is that you saw some very amazing results, but you also saw major safety challenges in the kids. I wonder if you could talk a little bit about what you learned there and that experience a little bit, because I think it was probably very formative how you think about drug development today.
Absolutely. I think that to this day, that has been one of the most rewarding and yet one of the most sobering and challenging lessons that I've gotten as a drug developer. So back in 2017, when we initiated the program to develop AT132, that is a gene replacement approach for X-linked myotubular myopathy, where we are delivering a healthy copy of the gene in pediatric patients. We had spent quite a bit of time with experts in the field to try to understand expectations that we would have in terms of the recovery that we could see in these patients. And just by way of an introduction into XLMTM, think that what you experience or what you observe in kids who are born with XLMTM is that the majority of them go rapidly into respiratory failure after birth, and the majority of them by 6 months of age require a tracheostomy, and they are dependent on mechanical ventilation that eventually becomes 24/7. They are unable to swallow many times, even a drop of water. So there's constant management of secretions, and many of them don't have any anti-gravity movement. So it's very, very challenging for these kids, and they are completely dependent, the majority of them, on their caregivers. Mortality associated with XLMTM to this day is about 50% by 18 months of age. And the ones who survive are dependent on the gastrostomy and the mechanical ventilation and all of that support. So when we talk to neurologists and pulmonologists and parents, I distinctly remember that some of the expectations of benefit were quite measured. So pulmonologists told us you can get a patient who has been 24/7 dependent on ventilation for 2 years. You perhaps can get them to reduce that by 10, 15 minutes a day. So and parents just wanted 5 minutes to give a bath to their kids, or to take them to the playground and just throw them down the slide, right? So the expectations initially were, again, quite measured. And to my surprise, I still remember that phone call in December 2017 when one of our investigators told us, hey, the kid who we administered the experimental drug to 3 months ago, who had no anti-gravity movement, is trying to pull off the trach from his neck. Say, what? I mean, not only did that mean that that kid had now enough strength to raise his arms and to try to pull it, but also it meant that he didn't need the requirement of mechanical ventilation as he did 3 months before. So what we witnessed over those 3 years was incredible. Many of these kids were able to meet milestones that had never ever been met by XLMTM patients. And many of those patients became ventilator-free, decannulated. And many of these kids ended up being able to walk and run. So it was incredible. We presented results at the World Muscle Society meeting in 2019 and at the American Society of Gene and Cell Therapy. Many journals were fighting over the data being published in the journals. It was incredible. And in early 2020, Audentes was acquired by Astellas Pharma. This came around the same time that I got my present opportunity at Encoded. And I made transition to Encoded, fully convinced that the XLMTM data would lead to a BLA approval within a few months or a year of my departure. So I couldn't have been more surprised by the knowledge that 4 months later, there were fatalities in that trial, with 4 kids unfortunately passing away due to hepatobiliary disease that had not previously been described in the disease that was exacerbated by the high viral dose and administration of the experimental therapy. So that, needless to say, was tragic. It was sobering. It was obviously traumatic for all of those families to observe this somewhat binary response, where the vast majority of the patients, of the 27 patients who were dosed, the vast majority of them saw an incredible benefit. But then you couldn't really predict who could have the complete opposite outcome.
Right.
So that has, on the one hand, given me a lot of humility as I think about rare disease drug development and about working at the forefront of science. But also, I think that has reiterated the importance of a very exquisite understanding of the natural history of the disorders that we're trying to treat.
Because I read the paper in the Lancet, and I think that one of the it was probably a discussion item at the time, but there was some data already that they were looking into, which is there was probably some subtle evidence in some of the cases of previous hepatic issues, and just to take a step back, there's often liver concerns in gene therapies because of the way the delivery works and the viral mechanism. This was somewhat different than other gene therapy cases where you see response from prednisone steroids that help with the immune response, right? So this was perhaps something that was latent in and related to the disease to begin with, but then is exacerbated by the therapy in a hard to predict way until afterwards, and you learn this very harsh lesson, right? And so there's one of the things that also struck me about this is how fragile these children are to begin with, right? So you see this binary outcome where some of them have incredible response, but others, there's just so much going on that it's really difficult to predict in some cases what a major therapeutic like this is actually going to do, right? And only after you see some of the data is it possible to appreciate just all the moving pieces, right, that's going on from a biological perspective inside these kids. Is that an accurate summary?
That is a very accurate summary. And, for the audience, I think that there are three distinct things, two of which we were aware, liver related, and one that is the thing that we unfortunately learned through the fatalities and the autopsy cases. We knew that patients living with XLMTM many of them had hepatic pili, and this was a risk that we try to mitigate by ensuring that we had clear ultrasounds, liver ultrasounds, and that we had liver function within certain limits of normality. We also were aware about the dose-dependent transient elevation of transaminases that has been associated with AAV gene therapy all the way from the hemophilia days, about more than a decade ago. What we didn't know is that these patients, in addition to the piliosis, they also had some of them had this proclivity for cholestatic liver disease. So that was what was surprising in the end, and that was what eventually led to the fatalities in association with the higher doses and in some of the older patients where when the systemic administration of this type of therapy was calculated by weight, so the total dose that some of these patients got was quite an important one.
Yeah, that makes sense. So is that something that I guess going forward, and maybe a lesson, well, certainly a lesson probably broader than XLMTM, is natural history studies or other ways to understand what is the complex set of comorbidities that's going on in these patients? Maybe you could talk a little bit about what you've learned about that, or how you're applying that today.
Yeah, absolutely. I think that spending enough time trying to understand the natural history of the disease is fundamental. Frankly, for XLMTM, we did a retrospective natural history study, a prospective natural history study, and this is something that we were not able to capture. So in that now what we try and do is we spend a little bit extra time trying to understand any potential patterns that look a little bit different in potential subpopulations of the diseases that we are trying to target. So I think that is something that we are doing now as we're working at Encoded on mental diseases such as Dravet syndrome. We also conduct a very, very thorough assessment of the natural history of this disease that we concluded a year and a half ago, and that we're utilizing as we are designing our clinical programs for this type of disease. So that's a fundamental piece that one has to do, and sometimes you just miss it, right? And I think that the other thing that is quite important is for us to act with a huge sense of urgency, but at the same time be very cautious. And I mean, that's why we stagger dosing in our early phase clinical trials. That's why there is a period of time that we wait in between patients so that we give time for protein expression and then the results of protein expression to be evident for you to understand the acute, subacute, and potential chronic toxicity of your drug. So definitely a lot of lessons learned that we continue to apply as we work on other disorders.
So what drew you to Encoded? I think it was 2020 that you said that you left. What got you interested in this probably very small company at the time, a bit bigger now, but what got you interested in it? I know I'll talk about what I found really interesting about it and why I want to talk to you in particular, just based on my research background, but I'm curious to hear from you first.
So the couple of things that attracted me about Encoded were that our founders we're already thinking about the next wave of gene therapy products that went beyond the gene replacement approach and that were a little bit more precise. So at Encoded, we have two platform technologies. On the one hand is the identification of novel regulatory elements to potentially constrain gene expression to certain cell types. Yeah. So we have a genomics-driven screening platform that is proprietary at Encoded that allows us to scan thousands of sequences of regulatory elements to try to understand the patterns of gene expression in target cells. Yeah. And then we apply machine learning algorithms to identify potential combinations of enhancers, promoters, 3' UTR regions to try to find the specific patterns of gene expression. And I thought that that was not only fascinating because that really makes precision medicine more precise, but I thought that that was a condition sine qua non. We could not really address some of the neurological disorders that affect certain cell types, where just the paneuronal expression may be deleterious from a safety perspective. So I thought that that was very, very novel. And then the other platform is that of the design of novel regulatory, novel transgenes with different functionalities. So for Dravet syndrome, for instance, since the SCN1A gene is the one that is mutated in 85% of the cases, is too large to fit into an AAV capsid. What our non-clinical team did is design an engineered transcription factor to upregulate expression of the gene, and I thought that was a very elegant approach. And now, for instance, for Angelman, we are developing microRNAs that knock down the expression of the antisense sequence and that naturally silences the expression of the paternal allele. So we are compensating for the loss of the maternal allele with unsilencing the paternal allele.
Because it's an imprinting disorder, right?
Yeah, exactly.
Interesting.
And this is similar to what antisense oligonucleotides do. It is just that we do it without one administration, right? So I thought that the combination of both those things was very elegant, very promising, and very exciting. So those were the two things that attracted me to it.
I think both those are super interesting. The first one was the thing that stood out to me in particular because I did my PhD work in a particular class of regulatory elements called ultra-conserved elements. So I was working in developmental disorders and trying to find non-coding causes. And these elements are parts of the genome that aren't genes, but they're likely enhancers or some other regulatory element. And the sequence is basically identical all the way back to mice or fish or even further beyond in evolutionary time. And so the hypothesis was clearly there's something important going on here. They're exquisitely controlling gene expression in some of these important genes. But more broadly, I think the thing that was very interesting to me through working on that project in the UK was how many haploinsufficient neurodevelopmental diseases there are. So these are cases where you lose one copy of the gene and you have, major issues in the child. And these are going to be challenging to treat. But one, I think, really exciting idea is, can you go to the regulatory element of the healthy copy, drive up expression, and restore it either in the whole body or potentially in the cell type of interest, right? And so I thought that the way you all were thinking about it and some other companies as well to do this kind of really precisely targeted regulatory element-driven approach was interesting, and it could actually be a way to bridge the gap towards common diseases in time, right? Because you could think about more precisely tuning gene expression in some way that isn't delivery of an entire gene, but is just changing the expression levels in some more tightly controlled way. I think we're probably thinking decades down the line here, but it's interesting to think about where the technology might go with time, right?
Yeah, absolutely. Hopefully not decades. Hopefully less than decades.
Yeah. I mean, what do you think? What needs to happen to pull us towards that future?
Boy, I think that's a tough question. I think that on the one hand, we need to do our homework with some of these monogenic disorders and show the potential transformative power that this approach has.
Yeah.
And then I think that we need to veer into some more common diseases that may be amenable for this fine tuning of expression and generate some proof of concept data. And I think that the combination of efficacy on obviously safety data in the monogenic disorders, plus some proof of concept in some larger disorders, I think that will rapidly unlock the possibilities of using this in other disorders, even when we're not talking about just a monogenic disorder, but more polygenic, because we're trying, as you well pointed out, we are more tightly controlling gene expression. So perhaps just a little bit would suffice to help in other disorders.
How much is delivery of the transgene or microRNA or whatever it is that you're working on a bottleneck in the work that you do? Because there's been a lot of work in different kinds of capsids, nanoparticles. I'm interested to get your take on what the unsolved problems are there still, and also where you're seeing some good progress.
So delivery is definitely an issue to contend with. We have tackled this from in a very scientific and dispassionate way. And a few years ago, back 5, 6 years ago, what the company did is conducted a route of administration study in non-human primates comparing the relative biodistribution of our candidate for Dravet syndrome, comparing IV administration with intrathecal lumbar, intrasternal magna, and intracerebroventricular, bilateral, and unilateral, and just assess the transduction, mRNA expression, and protein expression that resulted from all of those routes of administration. And what we learned through that is that the results did not vary at all between bilateral and unilateral ICV administration, and were not really different between ICV or ICM, but pragmatically, after interviewing several neurosurgeons, all of them were very familiar with ICV. Very few were familiar with ICM, route of administration. So that's what made us decide for ICV administration, because what we wanted is to start with the route of administration that allowed us to use the lowest possible dose from a safety perspective, and at the same time get us the best biodistribution. So that's the route of administration that we're using for our clinical development program for Dravet syndrome. That's also what we intend on using for our second program in Angelman syndrome. But I know that there are a lot of advances in the capsid world that may allow us to, at some point, transition into intravenously delivered capsids that get us the similar biodistribution or even higher than what we achieve going directly into the brain. And I think that obviously that is going to be a much more permissive route of administration that may enable us to also go back to your previous question and make these a little bit more amenable for other indications that are a lot more prevalent.
Yeah, that makes sense. How do you think about RNA medicines, gene therapy? Do you see one or the other as being something that's more likely to see major success in the long term, or do you think it's going to just really be dependent on the condition, and each of them are going to find their niche? Because that seems to be one of the big questions right now. Of if you look at, I don't know Dravet in much detail, but there are some people are using. You mentioned it for Angelman already. Some companies are using an RNA-based approach. Some are using a gene therapy-based approach. There's it's unclear, right, what is ultimately going to be the best route, whether you have one-time administration or infrequent annual or biannual doses. I'm just curious to get your perspective on those two, because they're very different ways of thinking, right? We go and address the RNA molecule, the DNA molecule has pretty profound downstream consequences.
I think that patients need more options. And I think that each one of these therapies will find a niche eventually. And I think that we can benefit in the gene therapy realm by some of the progress being made with some of the other approaches. And just to go back to the two first indications that we're tackling, Dravet syndrome and Angelman, in both of those indications, there's a precedent now in development in phase 3 in both cases, one for Dravet and two for Angelman syndrome, where there are companies developing antisense oligonucleotides that have proved the concept that upregulating the expression of the SCN1A gene has a therapeutic effect,
right,
that reduces the seizure burden, that improves development. And the same case in Angelman, there are two different antisense oligonucleotides that have shown improvement of development. And those at present are 4 time administrations annually, so every quarter. It's an intrathecal administration. I think that the results speak for themselves. I think I wish those companies the best because I fundamentally want those families and those patients to have better alternatives. And those learnings are helping the gene therapy world to also understand exactly how rapidly you can see potential improvement, how profound, and I think that eventually each one of these therapies will find a niche.
Yeah, that makes sense. You mentioned something interesting there, which was about learning from other peers, maybe in your same space. But you've also worked across infectious disease, psychiatric, and quite a few different therapy areas. So it strikes me that you have the opportunity to bring some transfer learnings from your past career as well. Are there things that you see, for example, the psychiatry community doing, or the infectious disease community doing, where when you look at gene therapy, you go, like, guys and gals, we really should be thinking about doing it this way because those people over there have figured this thing out that's just taken us a little while because the field has followed some path dependency. What springs to mind there?
Yeah, so one of the clear learnings for me, for instance, as I think about clinical outcome measures and things like how we do the clinical global assessment of improvement in these rare disease clinical trials, all of that work has been done for 3 decades in psychiatry, right? So back in the day when I was working in obsessive compulsive disorder, I worked with some of the people who actually came about with the concept of CGI, right? And how to train the investigators to do these in the same harmonized way, and how to develop and validate those instruments so that they can pass regulatory muster. So all of those learnings have served me very well as I try to select outcome measures from natural history studies and then try to validate them to be used in the interventional programs that we launch. That's just one, for instance, of the areas that has served me well from experience from more than a decade ago.
Yeah. What on the subject of natural history studies, what have you learned about there's this kind of interlocking set of challenges where, on the one hand, in your role, I think you need to design the most scientifically rigorous study, which may often mean, hey, I want to do a natural history study for years, collect lots of data points so I really understand the baseline. I want to design a clinical trial where I measure lots of things and make sure I'm really getting it. But that also creates patient and family burden, where the more things you're measuring, the more visits you're having, that has an impact on enrollment. How do you think about addressing all of these potentially competing priorities? There's a negotiation at the core of this, right? Where you have to run the most scientifically rigorous study, but notwithstanding all these other constraints that you have. I'm wondering if you have some thoughts on that.
Absolutely. I think that we have to keep an open mind. And a good example of a challenge that we faced, for instance, for the natural history study that we launched for Dravet syndrome back at the end of 2020, is that we wanted to collect a ton of data because there was not enough data on the trajectory of the seizure burden and neurodevelopment, especially in the youngest patients living with the diseases, and that had been contemporaneously generated. And so we decided to launch the study in the middle of the pandemic. And we had designed that study before the pandemic erupted. And when the pandemic initiated, we recognized that we need to be a lot more pragmatic, that we couldn't be bringing patients to hospitals that were full of COVID patients every 3 months. And we were pushed to find innovative ways, creative ways to decrease the burden on these patients, but still collect the data, because at the end of the day, the natural history data would not only allow us to design a better trial and argue for the inclusion-exclusion criteria that we would eventually propose in our interventional program, but also we were thinking that at some point, and we're still thinking that natural history study will serve as an external control. So one of the lessons learned that I've gotten from many years in industry is that we cannot be dogmatic, and we need to honor the commitment of patients who participate in our clinical studies by generating high quality data. Our science needs to be robust and needs to be interpretable, and it needs to be irrefutable, so that we have easier conversations with regulators, that we're not criticized for poor quality data. That is not acceptable. I don't think that we're doing a good service to the community if we don't introduce enough rigor. So it's very dependent on the indication and the precedents. But at the end of the day, I think that we do need to make the investment in those natural history studies so that we can get the learnings that we need for interventional studies, and that also we can potentially make use of those external controls. Because the reality is that there are a lot of examples of companies who have struggled because the external controls are not of adequate quality, right? So that's the tension.
That's the theme that we've discussed a number of times on this podcast. That seems to be really hitting home with rare disease focused companies. You invest in these high quality natural history studies, and the FDA really does value the data very highly if they've been designed correctly, right? And you're measuring the same kinds of things and it's done rigorously. If it's just a mess, then it's useless, right? Really, from that perspective. And I guess that's an important point for the patients and families that take part, right? If this isn't just a scientific curiosity, this is really going to form a core part of our package to make sure that this is safe and make sure that it works, right? Yeah. As we wrap up here, just a couple more questions to give people a sense of where you're heading with the Angelman and Dravet programs. What are you expecting, or what should people expect in the next couple of years? One of the challenges in this world is it does take years sometimes. You've talked about trying to bring that pace up, but what does great look like in the next couple of years if you're able to achieve it?
Well, I am incredibly excited about our Dravet syndrome program. First, because that's our lead program. We launched the first phase of our development program for Dravet syndrome last year, with 3 complementary open-label ascending dose trials where we're assessing the safety and efficacy of ETX-101 in patients under 7 years of age. And this is in Australia, the UK, and also the US. We have dosed several patients across several doses, and we'll be reporting data in the second half of this year. So I am incredibly excited about that. And incredibly excited that one of those trials, so first, we were able to consolidate the 3 complementary studies into a program that we call Polaris. So we got regulatory agencies in the 3 countries comfortable with the idea of cross-referencing the safety data between each one of the studies, so that we didn't have redundant dose exploration, but at the same time we were able to proceed more rapidly with enrollment in the program. So that data, I think that will be quite robust, and it'll serve us to transition into the second part of our program that is not open label. It introduces all the methodological rigor that regulators require, including blinding, sham control, long duration of observation. So I'm excited about that transition, but I'm a dreamer. So I like to dream that the data that we disclose at the end of this year is so robust that we can go back to regulators and argue for perhaps less burden for our patients participating in the trial, or perhaps an accelerated path to a potential approval, so that these potentially transformative therapies reach patients more rapidly. So that's what my dream is for Dravet. For Angelman, we plan to file a regulatory submission sometime within the next year or so, and I'm quite excited about that. I think that what the antisense oligonucleotides have shown is that you can see an improvement across different domains, including communication, cognition, and some of the behavior. And I think that I'm excited about what we can do with a one-time administration in this field. So that is what's going to happen over the next 2-3 years, and I'm quite excited, and I feel blessed to be part of it.
That's amazing. I think that's a really exciting note to end on. If people want to help, if they want to join the mission, are you hiring? What would be most helpful for people? And they can keep track of you at your website. It's encoded.com, which is a great domain. Someone did a really good job finding and securing that domain. But what's most helpful for you from our listeners?
So definitely they can find information on what we're doing at our website. We also have a LinkedIn channel, and sometimes we post the things in YouTube of the progress. We're going to be presenting data at different congresses this year. We have been working with the clinical and the patient community for years now. So I think that there's a lot of excitement about what we're doing. We're excited about partnering with people on this effort. So if there's anyone interested, please reach out to us. And stay tuned. And if you see me in a meeting, please come and say hi.
Wonderful. Well, thank you, Sal. I really appreciate you taking the time. I think you shared a ton of lessons here today, and you've been doing some amazing work for patients over the course of your career. So best wishes to you and the team for the exciting milestones this year. I'll be keeping an eye out as well. And hopefully you get that amazing data that you're hoping to see, and we'll be off to the races helping patients.
Thank you so much, Patrick. It's been a pleasure.
Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.